نعم. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. الحمد لله والصلاة والسلام على رسول الله أما بعد. So in our last class we discussed cell division. We started with sexual and asexual reproduction, and we're trying to go through how all these processes are in the lower animals, the invertebrates and the vertebrates. And then we stopped at cell division. We're trying to discuss and understand mitosis and then meiosis. So in order to just recap on cell division, I said cell division is a process by which a cell divides to form new cells. Not necessarily two cells, because we know we have multiple fission, if you could remember, which is done by amoeba and divides into not just two cells, multiple cells. So, but most times it's usually binary. That's why the definition here says two new cells. So in the cell division in prokaryotes is what is called binary fission. And remember, I told you to understand the difference between fission and fusion. So when I say nuclear fission, is different from nuclear fusion. Fission means to divide, fusion means to join. So in prokaryotes, example bacteria, yes. So you know prokaryotes in kingdom prokaryotes, in kingdom Monera, they are prokaryotes. Those are the only prokaryotes we have. And then we have schizophyta, which are the bacteria, and the cyanobacteria, which are the blue-green algae. So it's binary fission, and we already explained what binary fission is. The cell does divide into two, and then you know how it divides: the nuclear division, the cytoplasmic division, and that's all. It forms two daughter cells, and the daughter cells are very identical. Now in eukaryotes, we have two types of division. The first one is mitosis, while the second one is meiosis, but they are for different purposes. Mitosis is a division, a type of division that is meant for 1. Growth, 2. Repair of worn-out tissues or healing of damaged tissues, and 3. It is as asexual reproduction in some animals. That is mitosis. While meiosis is a division that is meant for the formation of gametes only. A division that's meant for the formation of gametes, that is sex cells, the sperm and the ova. So that is meiosis for you. Yes. And then in order to understand this, so you say mitosis is for somatic cells. Yes, mitosis is for somatic cells, while meiosis is for germ cells. So if you try to remember, I gave you the definition of somatic cell and germ cells. Yes, those are the two different types of cells that we have. Let me define them again. Somatic cells are body cells. When you hear somatic, it's body cells. Yes, even though germ cells are still part of the body, but somatic cells are body cells. They are cells in whose genes will not be passed on to the future generation. Yes, they don't partake in anything sexual reproduction. You don't copy their genes into the next generation. They are just the body cells, other body cells. While when we talk about germ cells, these are sex cells, cells that are destined to the cells that are destined to become gametes in the future. A cell that is destined to become gametes is called a germ cell. Yes, so if it will later become sperm or egg, it will be called what? A germ cell. So these germ cells, they are cells whose genes will be passed on to the future generation. So that's the difference between the somatic cells and the germ cells. So that is the mitosis, difference between mitosis and meiosis. So let's briefly recap mitosis, even though we've done it in the last class. But don't forget, I said mitosis is for somatic cells and is for growth, the repair of worn-out tissues, and asexual reproduction. Yes, so when the cell grows or any tissue or cell that is damaged, it's the mitosis is what takes place there to repair it. Then asexual reproduction, just like we've explained, mitosis, asexual reproduction in different animals. So while meiosis is for germ cells, and then it is for formation of gametes, which are sex cells. So remember that. Now, um, so we talk about this, the major steps, the DNA, the nuclear division, then the cytoplasmic division. So let's go to mitosis proper. Now, in mitosis, we have what we call the cell cycle. We have what we call the cell cycle. So basically, it is the these are this is the process in which the cell undergoes throughout its life till it dies. So from the beginning, what it is, what it is, what it is. You can guess what the cell cycle is. You can pause the video and guess to try to see if you really know what the cell cycle is. But if you don't know it, probably you should go back and collect your school fees from your school. So you'll be taught in SS1 the cell cycle. Now, talk about the cell cycle. These are sequences of events from the time a cell arises as a result of cell division. Yes, after cell division, that cell that is formed until the time the cell divides itself. Do you understand? When it divides again, the cell it gives rise to will now go to the cell cycle again till the time. So that's it. So cell division, the after cell division, a new cycle will begin. Do you understand? So cell cycle basically consists of stages, and these are what? Interphase, that's the first one, and mitotic phase, that's the actively dividing phase. You know, when we always talk about why we always separate interphases, because there is no actively division, there's no any active division in interphase. So which you usually call the resting phase, even though we do say it's not the resting phase. But then we have the interphase and then the mitotic phase. So the interphase consists of three different phases. The interphase consists of three different phases. The first one is the G1, the second one is the S phase, and the third one is the G2, as you can see on my screen. So we have the G1, the S phase, and the G2. Cells spend most of their lifetime in interphase, as you can see. It takes most of the last, and the most part of interphase is the G1. Don't mind me, so you are saying S is bigger than G, right? But it is the G1 that is what, that is actually the phase in which the cell spend most of its time there. Now let's talk about the interphase, the G1. In the G1 phase, what does the cell does in the G1? So what happens in G1 phase? One, the cell increases in size. It absorbs nutrients. It takes in, thus basically what happens in the G1 phase is growth. Yes. So that's just this G for growth. You understand? G for what? Growth. So what happens is growth. And for growth to happen, you absorb nutrients, you increase in size, you increase in mass. So that is basically what happens in G1. Why in S, that is called the synthesis phase. The synthesis. So just like you said, nothing is not that cell is actually resting. The meaning of that resting is that it is not dividing. Do you understand? But it consists of three phases. The first one, so after it's finished eating, increasing in size, so it moves to the S phase. So when it moves to the S phase, as you can see in interphase, you can see the chromosomes. They appear like what? Like chromatin granules. So as even you can see like this, they are not, you can't differentiate, they are not differentiable under microscope. So you can't really see the chromosomes. You just see strands of what? Chromatin. So, and these are centrioles. Do you understand? So it's okay. I'll still explain this. This is the nuclear membrane, and this is the nucleolus here, and this is the nuclear pore. This is the chromatin. Do you understand? It's chromatin, not chromosomes. So it's chromosomes that is present in the structure of what, in the structure of chromatin. Yes, the chromatin will be the one to form the chromosome, of course, definitely. So in S phase, what really happens in synthesis phase is this chromatin will replicate. That's basically what happens. The chromosome, the chromatin will replicate. Do you understand? So which means the DNA that is there too, the chromosome that is there will duplicate. So that's basically what happens in the S phase. Then the G phase, the G2 phase, which is the second growth, what will really happen is all other structures that are needed for mitosis will be formed, because after G2 phase, remember, after G2 phase, it is going to enter mitotic division straight. So it prepares itself at this stage for mitosis, whatever it is that will happen. So it is at this phase that the centrioles that is inside the animal cells, the centrioles divide. Do you understand? And any other structure that will be needed for mitosis will be formed at this stage, at the G2 phase. So basically, at interphase, what will really happen is that cell will grow. Of course, it has been growing the first part of interphase in its life. So the first part, the reason why it will grow is because this is immediately after the cell division. So immediately the cell is being formed, it is a small cell. So at G1 phase, it will be growing, it will be growing. At S2 phase, when it's moving closer to when it wants to divide, so it's what replicates the chromatin duplicates. And when it reaches the G2 phase, what will happen in the G2 phase is that it actively prepares itself for mitotic division. So the centrioles will divide, and all other structure that will be needed for mitosis will also be formed. Now, this is a structure. This is how the chromosome looks like. As you can see, these are sister chromatids, just like we explained the other time. So sister chromatid. So it, because when it duplicates, we can now have, when it duplicates, the chromatin, remember in interphase, it duplicates, and once it duplicates, it means once the chromatin forms, condense to form chromosome in the prophase, we can now have double structure. Like you cannot have them in pairs, you cannot have the chromatin in pairs because they've replicated. So this, you can see, is a shorter arm that is called the P arm. This is the longer arm that is called the Q arm, and this, the center, is called the centromere. You understand? So sister chromatid are being held together at the what? At the center. So it's not just center, like this place is the same with this place. No, this place is shorter. The upper hand is the shorter arm called the P arm, while the lower arm is the longer arm that is called the Q arm. So it's basically something. So we've explained cell cycle. So it's basically something like this. So just this is the structure again. As you can see, G1, the S phase, G2, then it enters mitosis. So the mitotic division itself, that's what really happens in mitosis. Just like we explained in binary fission, it's just the division of nucleus into two and the division of cytoplasm into two. So the first one is what? Nuclear division and cytoplasmic division. That's just what happened to form new daughter cells. So we have the mitotic division. So we have the after the interphase, which is the resting phase, we have the prophase, the metaphase, anaphase, telophase, and then the last one is what? Cytokinesis. So once the cell enters the first stage, which is the prophase, this is actually the interphase, as you can see. So I've corrected this. Is chromatid, not chromosome, as you can see the other time. This is central. So this is how the cell, just understand how the cell looks like in interphase. Now, once you enter prophase, what happen? The first thing that will happen in prophase, nucleus will disappear. Do you understand? Nuclear membrane will also will disappear. But you know, I told you not to be always saying disappear, right? So it disintegrates, not disappears. So it disintegrates. You understand? But even though if you say disappear, normal, but talk like biology students. So it disintegrates. Then nuclear membrane also what? Disintegrates. What do we have again? The centrioles start moving to the opposite pole and spindles form. Then lastly, the chromatin that is present will need to coil, condense, and shorten to form what? Chromosome. So it's something like this. Just can you see? So mitosis is about to start, and this one is like telling the chromatin, dude, what far now? We are still not condensed. How far? You need to condense to àti fẹ́ bẹ̀rẹ̀ mitosis àbí ṣé ń sùn ni? Ẹ má sùn lọ. Do you understand? So that's what will happen. So that's just what will happen in prophase. So remember what will happen after you've seen how it is in interphase. Remember what happens in interphase. So the centrioles divide from its activity take in whatever it is in G1 phase. In synthesis phase, this duplicates, the chromatin is duplicates. Then in G2 phase, all other annual that are everything that every structure that is required for what's it called for mitosis is formed. Then centrioles divide into two. Now, prophase, we said nucleus disintegrates. That's first thing. Nuclear membrane to what? Disintegrates. Spindle form when the centrioles move to the opposite pole of the cell. So that's basically what. And then chromatin what coil, condense, and shorten to form chromosome. And this chromosome is represented by two sister chromatids that are joined together, connected at the center, that is called the centromere. So this is just late prophase. This is just what I just that we just understood what happens in prophase. Now in metaphase, we move to the second stage after everything has happened in prophase. I'm just rushing it because I've discussed it before. In metaphase, just like the name implies, the chromosomes align themselves as at the metaplates or the metaphase plates or the equator, whatever it is you call it. So this is basically what happened. In prophase, we said the centriole starts moving to the opposite pole, but metaphase, they've reached the opposite pole already before they enter metaphase. So as you can see at late prophase, they have already reached the opposite pole, and you can see there is no nuclear membrane again. There is no nucleus, and you can see the chromatin have become chromosomes, and you can see them at the center. They are still at the center though, but even though there's no covering for them. So at metaphase, what will happen is that all the chromosomes will align at the equator. As you can see them, they align at the equator, or you can call this place the metaplates or the metaphase splits. And each chromosome is attached to the spindle fiber at its center, the centromere. That's just what happens in metaphase. That's all. That's all what happens in metaphase. That's all. Now, then the next stage is the anaphase. Once they've attached, what will happen? At interphase, the spindle fiber begins to what? Pull the chromosome as to the opposite part of the cell. So they separate the chromosome, they divide them. So those pair chromosomes, that's those sister chromatids, will now divide. And I told you, once sister chromatids divide, they are called what? Daughter chromosomes. They become daughter chromosomes, isn't it? So each one will be called daughter chromosome. So at anaphase, they are being pulled to the opposite pole of the what's it called? They are being pulled to the opposite pole of the cell, and that's just what happened at anaphase. And at anaphase, if you look at the cell, you can see that something is at this stage and at this place, like it seems the cell is trying to go inside. Yes, so that's the furrow that starts in this one. Cytokinesis. Cytokinesis does not wait for everything to happen first, so it starts little by little, little by little. So we move to telophase. As you can see at telophase, they've already reached their destination. The nucleus comes back. That's one thing. The chromosomes uncoil, do you understand, and letting to form to form the chromatin back. The nuclear envelope, that's the nuclear membrane, form back. The spindle fiber disappears, and then the nucleus comes back too. And as you can see, cytokinesis is setting in. So this is the furrow that's happening. Cytokinesis is already setting in, and that's telophase. That's the last phase. So basically now, after everything has happened in telophase, cytokinesis will now happen complete. Cytokinesis will now be complete. So cytokinesis is just the division of the cytoplasm, and this only happens in what? In animal cell, because of the cell wall is not the cell membrane is not rigid as the cell wall in plant cell. So furrows form like this. Furrow forms like this. The furrow will form and deepen inside this, and then it will separate it. But if it's in plant cell, I told you what will happen. Cell plates will be formed, and these cell plates are small vesicles that are being produced by the Golgi apparatus that will line up and they fuse together in order to divide the cell into two. So that is the end of mitosis. That's just mitosis for you. Yes. So as you can see, so you can pause the video and try to look at it and name it yourself. Which stage, and look at it. Try to understand different structure at different stages. Try to understand what they represent at different stages. And look at this again. Tell which stage is this. Ask yourself which stage is this. Pause the video and try to observe it. Which stage is this? Then identify this X, identify Z, identify Y, and identify W. So identify all those things, and that's self quiz. So which phase is this? Name this phase too. Okay. Oh, sorry, I've already named it. It's telophase. All right. So it is only at telophase that they've already reached the end of the pole that the spindle fibers were drawing them. So that is all. So if you have any more, so after all the mitosis, whatever it is, there is another stage that is called the apoptosis, which is cell death. So definitely everything will die. Kullu man alayha fan, isn't it? Kullu nafsin dha'iqatul maut. So everything will die, of course. So our cell death is called apoptosis. So you can go and read more about it. So that's just all about mitosis. So in my next class, we'll talk about meiosis. If you have any question, don't hesitate to reach out to me. سبحانك اللهم بحمدك أشهد أن لا إله إلا أنت أستغفرك وأتوب إليك. السلام عليكم ورحمة الله وبركاته.